answer your questions.
Hello, my name is Christina Simonyan, and I will discuss the potential clinical implications of recent imaging studies in dystonia. The current knowledge suggests that the pathophysiology of dystonia is multifactorial. As Dr. Zak discussed in his talk, several genetic factors contribute to the development of this disorder and may be powerful targets for therapeutic developments. These genetic factors interact and in some cases define neural alterations in dystonia and may be influenced by several form-specific extrinsic triggers. The understanding of the neural underpinnings of dystonia has considerably evolved in the past decade. One of the first papers pointing to basal ganglia changes in dystonic patients dates back to 1949, where dystonia was referred to as a clinical concept, but the basal ganglia pathology with mineral accumulations has been outlined. Over the next several decades, other case studies and research reports pointed to additional evidence of basal ganglia involvement, culminating in the landmark paper of 1985 by Marsan and colleagues, which put forward the notion that dystonia is a basal ganglia disorder. Much research in the coming years followed with the focus on the understanding the role of the basal ganglia in dystonia pathophysiology. According to the basal ganglia model of dystonia, an imbalance of the direct and indirect pathways has been thought to underlie bottom-up abnormal decreases and intracortical inhibition and subsequently abnormal increases of motor cortical excitability. However, owning the significant advances and breakthroughs in visualizing the structure and function of the human brain, neuroimaging studies have started unveiling more complex brain disorganization in dystonia, involving not only the basal ganglia and thalamus, but also several cortical areas. Among the first studies in the mid-1990s, Eidelberg's group has systematically examined the glucose metabolism in cortical and subcortical brain regions in dystonic patients and determined a wider range of abnormal interactions, forming a neural network model of dystonia pathophysiology. In parallel, another model of dystonia focused on abnormalities in the cerebellum, as another target of frequent lesions leading to dystonia. This model proposed that lesions in the cerebellum may be causal to the pathophysiology of dystonia. As studies continue to be done in parallel, examining the basal ganglia, the cerebellar or neural network models of dystonia, both literature reviews and more analytically sophisticated research emerged, heavily weighing toward the network model of disease. And that's where we are at the current, where as of July, there are more papers being published that consider this disorder being network disorder rather than a basal ganglia disorder. Independent of the model, network, cerebellar, or basal ganglia disorder model, basal ganglia, thalamus, and cerebellum are being placed at the core of network disorganization, with the common features of dystonia being considered: loss of surround inhibition, abnormal plasticity of motor cortex, and abnormal sensory function. In addition, different forms of dystonia have been characterized by similarly widespread reorganization of cortical regions and their white matter pathways responsible for multisensory processing, sensory motor integration, and motor execution. Specific genetic mutations, polygenic risk, and extrinsic risk have been associated with sensory motor and parietal regional abnormalities. And several of these regions have been also associated with clinical symptomatology across the wide range of dystonic spectrum, while others have been implicated in endophenotypic traits of this disorder. Interestingly, task-specific dystonias, such as laryngeal and hand dystonias, were characterized by significant alterations in cortical areas compared to prevalently subcortical changes in non-task-specific dystonias, such as cervical dystonia and blepharospasm. In a more detailed overview of functional neuroimaging alterations in dystonia, these are typically characterized by abnormally increased brain activity, but decreased regional connectivity, as well as the disorganization of large-scale neural communities and the hierarchy that is necessary for normal information processing. Collectively, the studies significantly expanded our understanding of the functional changes in dystonia pathophysiology and started leading the way toward more targeted therapeutic options in these patients. The clinical implications of dystonia as a functional network disorder may be extremely impactful. Currently, the mainstream treatment is primarily geared towards symptom management, with the first line of treatment being botulinum toxin injections into the affected muscles. A recent study has estimated that nearly 40% of patients with focal dystonia received no treatment, and several studies urged the development of pathophysiologically defined treatments. Understanding the functional networks in dystonia presents a window of opportunity for the development of novel interventions that selectively target and modulate pathophysiologically abnormal functional neural network. One of the examples I'd like to review here is sodium oxybate, which is a centrally acting gamma hydroxybutyric acid that mimics the effects of alcohol. It is important to mention that up to 55% of patients with dystonia report improvement of symptoms after alcohol intake. Our studies show that the drug action is specific to brain regions altered in patients with laryngeal dystonia, with greater normalization of function seen in responders, as seen here in green, compared to non-responders in purple. These changes corresponded to substantial improvement of patient symptoms, as you can see in this bar graph, as well as you can hear in these two videos of a patient with abductor form of laryngeal dystonia before drug intake and 40 minutes after drug intake.
He is hiding behind the house.
He is hiding behind the house.
Hurry, Helen is far ahead.
Hurry, Helen is far ahead.
He has gone home because he is hungry.
He has gone home because he is hungry.
I have a friend whose home is a lighthouse.
I have a friend whose home is a lighthouse.
I hope you could appreciate and hear the difficulties the patient was having on voiceless consonant like H's during production of the sentences. Here's the same patient 40 minutes after intake.
He is hiding behind the house.
He is hiding behind the house.
Hurry, Helen is far ahead.
Hurry, Helen is far ahead.
He has gone home because he is hungry.
He has gone home because he is hungry.
I have a friend whose home is a lighthouse.
I have a friend whose home is a lighthouse.
As you can hear, he has very few, if any, breaks and not much of trouble with the effort completing the same sentences. Looking further into the dystonic network in these patients, we can clearly see that sodium oxybate does act at the dystonic network level, normalizing not only regional function, but also interregional connectivity in responders compared to non-responders. Here you see the purple regions that are present in both responders and non-responders and that are being actively modulated by sodium oxybate. However, responders have additionally modulated areas within the abnormal network, and these are represented with green circles, and you can see that their connectivity is much more enhanced than in non-responders. Another example of the clinical implications of considering dystonia as a functional network disorder is the emerging modulation of parietal premotor connectivity. Moving away from the pure motor causes of symptom generation, recent studies have pointed to parietal premotor alterations as a potentially important player in dystonia pathophysiology. It has been shown that the abnormal network may be instigated by the increased excitatory influence of the left parietal cortex onto the putamen, as well as increased right to left influence of premotor cortex. As I mentioned earlier, genetically vulnerable parietal premotor connectivity was also linked to the overall heritability of dystonia. And as a potentially powerful therapeutic approach, a recent study used continuous theta burst stimulation to influence motor cortical excitability through transiently decreasing parietal premotor excitability. This is a promising approach, and clinical implications of this need to be still explored, but this is the right direction of moving with the research targeting networks rather than specific brain regions. Moving on to the abnormal structure in dystonia, several studies have shown mostly increased gray matter volume and cortical thickness, and mostly decreased white matter integrity across different forms of dystonia. Genes and clinical phenotypes have been reported to impact the extent of these abnormalities within the sensory motor dystonic network. Collectively, these studies agreed that different forms of dystonia share similar structural abnormalities, and they also involve form-specific alterations in the regions relevant to altered motor control. For example, the hand versus larynx region of the motor cortex in hand dystonia versus laryngeal dystonia, respectively. The clinical implications of dystonia as a structural network disorder have been recently explored in terms of diagnostic biomarker development. As you know, dystonia is considered the third common movement disorder after essential tremor and Parkinson's disease. However, up to half of the cases are typically misdiagnosed at the first encounter, so its incidence may be underestimated. This is partly due to the absence of a biomarker as an indicator of the common pathophysiological